Our first leech of the trip, first terrestrial leech. There we go. When you go out into a forest in Southeast Asia, it can be very difficult to see mammals. But there are several ways to find out what mammals are in a given area. You can go and look for footprints or hairs. It's very difficult and time-consuming. Or you can set up a camera trap, but probably you want to leave it there for several months, maybe even a year. Turns out what we're able to do is go to a forest just for a day, collect leeches off the ground, collect them off of ourselves, sequence the DNA from their gut contents, and use that DNA to figure out what mammals are in an area very efficiently. Conservation biologists have already started to use these methods, but our results will help them determine how many leech species to look at and how many individuals of each species to look at to maximize the number of mammals they can find in an area.
The idea that you can get what we call IDNA from leeches that have fed on mammals or other rare species in an area that's under conservation threat is something that has developed in the last 5 to 10 years or so.
And into the bag you go.
We had about 3 weeks to a month to work in Cambodia. We went to 3 basic areas. One is Prevahia, which is in the north and getting up towards the Lao border. Then we were down in the Cardamom Mountains. This is the highest elevation that you find in Cambodia. And we find that there's usually a greater diversity when you have an altitudinal gradient. And then finally we went to Sema, which is a protected forest that has recently had tigers and elephants and so forth over by the Vietnamese border. And by doing all of that, we hope to capture the greatest diversity across the country.
First blood on the trip from Lily. How's your first leech bite?
I don't feel it.
You don't feel it?
Doing field work and having the opportunity to collect these organisms in their natural environment allows us to reconnect with the goals that we have as scientists of conserving diversity and why we study the things that we do. And it's also a necessary part of doing this type of research. When the leeches were present in large numbers, it didn't take long for us to spot them, and it didn't take long for them to find us. And so the collecting was actually quite abundant, and it was quite successful. In order for collection data to be useful for our research and for future research, we really want to, as accurately and as detailed as possible, record the location where we collected these animals. That includes, when possible, GPS coordinates, the date when the individual was collected, or any kind of environmental factors that would influence the presence of that specimen when it was collected. If anyone else in the future wants to go back and revisit a location in order to find the same species or to understand which mammals were present, that person can find the location where we were collecting.
The DNA so far have been pulling out a number of different animals. We've been finding bats, we've been finding civets, we've been finding barking deer, mouse deer, really tiny deer. And interestingly, we've started to even find birds, which are very rarely reported to have leeches actually feeding on them.
The 2015 Niarchos Expedition to Cambodia allows us to do the sorts of things that we do best as biologists, and that is not just to know the diversity, but to see them in context. When you find this species of leech or that species of leech, is it in a bamboo forest or is it in a hardwood forest? Is it at this altitude or that altitude? Was it a rainy day? Was it a sunny day? And really get the context of discovery associated with the specimens that are right here in the museum.
Leeches are certainly an overlooked organism. Very few people have studied them. There's very little known about them. Even though it's not a huge group with about 700 described species, there have only been a couple people who have dedicated their profession to studying these organisms, and there's a lot to be discovered on all levels.